Enzymes are known as biological catalysts. This is because enzymes are able to speed up specific reactions in the body. Enzymes are formed by amino acid chains folding and coming together. This means that enzymes are proteins. We need to understand how an enzyme can speed up the rate of a reaction. First of all, enzymes have an area known as the active site. This active site is unique for each enzyme. The active site is able to interact with substrates. However, because the active site of each enzyme is specific, only one reactant, otherwise known as a substrate, will be able to interact with the enzyme. This brings about the lock and key theory of enzymes. This theory suggests how enzymes work as biological catalysts. A specific substrate will fit into the active site of an enzyme. Once the substrate fits in, a reaction occurs and the substrate is broken down into two products. Also, the reverse can happen. So two specific substrates can combine in the active site of an enzyme to form one product. Enzymes are closely linked to a person's metabolism. Metabolism is the sum of all the reactions taking place in the body. The metabolic reactions taking place can come in three different types. The first is when a large molecule is broken down into smaller ones. The next is when smaller molecules are used to form larger ones. And the final type is when a molecule is converted into a different type of molecule. Enzymes having specific active sites is particularly helpful here, as they make sure all the different reactions taking place do not interfere with each other. Enzymes also make sure that the reactions take place quick enough for you to survive. We are now going to look at how temperature and pH affect the activity of enzymes. As you would expect, if you increase the temperature at which an enzyme-controlled reaction is taking place, the reaction rate will increase. This is due to the enzymes and substrates having more energy, and so they are more likely to collide at higher temperatures. This works for temperatures below 37 degrees for enzymes in the body. The temperature at which the reaction rate is highest is known as the optimum. Above this temperature, the enzymes become denatured. Denatured means the chains of amino acids begin to unfold, and therefore the shape of the active site changes. This occurs due to the bonds holding the enzyme together being affected by the high temperature. As a result, the substrate molecule will no longer fit into the active site of the enzyme, and the rate of the reaction will start to decrease. Once all the enzymes have been denatured, the reaction rate will be zero. Enzymes do not denature at cold temperatures, they just become inactive. It is also worth mentioning that the optimum temperature for enzymes can vary depending on their environment. For instance, some enzymes in hot conditions may work best at 80 degrees. Changes in pH affect the bonds holding the enzyme together. If a change in pH occurs, the bonds are affected and the shape of the enzyme changes. Again, enzymes have different optimum pH values. The next required practical is to do with enzymes and pH. You are investigating how the pH value can affect the breaking down of starch by the enzyme amylase. For this experiment you will have three different pH buffered solutions, test tubes of starch and a test tube of amylase in a water bath. You will then need to add a drop of iodine solution into each section of a spotting tile. In one of the tiles you should also add a drop of starch. Then, once all the test tubes have reached the same temperature, a sample of one of the pH buffered solutions is added to one of the test tubes of starch. Once stirred, amylase can also be added. At the same time, the stopwatch should start. After every 10 seconds have passed, a drop of the solution should be added to the next tile of iodine. This can be repeated for each pH solution. Iodine turns blue-black in the presence of starch, but stays brown when all the starch has been broken down. You should see the progression from blue-black to brown the longer the reaction is left for. The optimum pH solution can be seen by looking at how quickly the iodine changes using each solution. We are now going to look at the enzymes in the digestive system. Digestive enzymes are produced by glands and work outside cells to digest insoluble food molecules. We are going to look at how carbohydrates, proteins and fats are digested. Carbohydrates are made up of sugar units. Simple carbohydrates only contain 1 or 2 sugar units, while complex carbohydrates contain a long chain of them. Glucose is a simple carbohydrate, while starch is a complex one. Carbohydrases are the enzymes which can break down carbohydrates to simple sugars. Amylase is a carbohydrate and it breaks down starch. The digestion of carbohydrates occurs in the mouth and the small intestine. Amylase is produced by the salivary glands and the pancreas. Proteins are made up of carbon, hydrogen, oxygen and nitrogen. Proteins are needed for producing cells and they act as hormones, enzymes and antibodies. Amino acid chains make up proteins and different arrangements give specific proteins. These chains are folded and form 3D structures. In order to make specific proteins needed by the body, the proteins found in food need to be broken down into amino acids. Protease enzymes catalyse the breaking down of proteins. These enzymes are produced by the small intestine, the pancreas and the stomach. Lipids are fats and oils. They are very useful as energy stores for the body, and they are made up of carbon, hydrogen and oxygen. They are also insoluble in water. They are incorporated into the cell membranes of cells and hormones. This is a lipid molecule. It is a glycerol attached to 3 fatty acids. When lipids are digested, they are broken down to these individual components. The reaction is catalysed by lipase, and these are produced in the pancreas and small intestine. The next required practical is to do a testing for these 3 compounds. Carbohydrates has 2 different tests. If testing for starch, then add a drop of starch to some iodine. The iodine should go from yellow to blue-black. If testing for other sugars, Benedict's reagent is used. A test tube containing the sample and some Benedict's reagent is heated in a water bath. If the solution turns from blue to brick red, a sugar is present. To test for proteins, add the sample, some Biuret solution A and Biuret solution B into a test tube and shake gently. If the colour changes from blue to purple, protein is present. And finally, to test for lipids, add the sample, some distilled water and some ethanol to the test tube. A cloudy white layer is formed if lipids are present. We are now going to quickly look at the changing conditions of the digestive system. The pH of the digestive system is not constant. In the stomach, pepsin, which is a protease enzyme, works best in acidic conditions. So, glands in the stomach secrete hydrochloric acid to allow the pepsin to work effectively. The acid also kills bacteria found within the food. To protect the stomach lining from the acid, mucus is produced. The food will move to the small intestines after the stomach. Here, enzymes from the small intestine and the pancreas digest the food. But these enzymes need alkaline conditions. To turn the conditions from acidic to alkaline, the liver produces bile. Bile is an alkaline liquid and it is stored in the gallbladder. When needed, it passes through the bile duct and neutralises food as it leaves the stomach. Fats are insoluble, and so during digestion they stay as large substances. This is an issue as enzyme activity increases with surface area. So bile also emulsifies fats to form smaller droplets, which increases their surface area.